Hello, welcome to MLSQR Medical Lab Science Quick Review. Today, let's look into the basics of neutrophil function. Our questions and answers are similar to the ones in the study guide. As you may already know, the neutrophils are also called PMNs, polys, segs, segmented neutrophils, neutrophilic granulocytes. Reference ranges and units of measurement are given. They are made in the bone marrow during hematopoiesis, maturing from stem cells, pluripotential hematopoietic stem cells, common myeloid progenitors, to myeloblasts, promyelocytes, metamyelocytes, bands, to finally mature to segmented neutrophils. Mature neutrophils circulate in the blood 5 to 7 days, rolling and sniffing for chemical messengers until they get a signal to come into action. Stressors like infection and inflammation will initiate a catecholamine response from the adrenal glands so that we have a fight or flight response. And this will cause demargination of neutrophils from the blood vessels to the peripheral circulation where they are needed the most. Demargination of immature myelos, metas, and bands into the peripheral circulation causes a left shift, which appears like the maturity of the neutrophil shifted left. You'll see this with leukocytosis, and this goes along with the fever, and that is consistent with a bacterial infection. These chemical messengers are cytokines. Different types of cytokines play different roles in the immune system regulation and growth. Which of the following are true statements about phagocytosis? Not all leukocytes participate in phagocytosis, such as the lymphs and basophils, certainly not red blood cells and platelets. Let's see how neutrophils perform this vital function.
Ouch!
Neutrophils are not lone soldiers in the fight against infections. Let's see how the neutrophils work with other leukocytes, like a coordinated immune defense. When you get cut, the bacteria passes through the first line of defense, your skin, and resident macrophages senses the foreign invader and processes the antigens to present to other cells of the immune system. If it is a viral infection, star-shaped dendritic cells in the tissue processes the foreign antigens to secrete interferon, which interferes with viral replication and initiate the immune system by activating T cells. The major histocompatibility complex 1 and 2 are part of the gene complex which is used by T helpers, natural killers, and cytotoxic limbs to recognize foreign fragmented proteins,
Hmm.
further activating other immune cells by sounding the alarm. Killer T cells are dispatched to the battle arena. Cytotoxic T limbs have weapons called perforin and granzyme B for the infected cells to commit suicide by apoptosis. Complement activation plays an important role in opsonization, which coats the bacteria like butter, making it ready for the phagocytes to consume them. They recognize the opsonyms like C3b. Also, complement has a membrane attack, which may kill the invader directly. Natural killer T cells are also activated by lipopolysaccharides of the bacterial cell membrane to secrete interferon, which hyperactivates the macrophages. So far, we've seen the innate immune system work in a quick, nonspecific response to foreign invaders. The specific immune response involves B cells with immunological memory to make specific antibodies against the foreign antigens, providing a delayed but stronger and more robust specific immune response. Activated macrophages secrete tumor necrosis factor and interleukins. The neutrophils roles in the capillary, sniffing for inflammatory signals such as complement frags, tumor necrosis factor, interleukins, and lipopolysaccharides, and FMET peptides from the bacteria. The neutrophils interacts with ICAM and selectin adhesion proteins in the inflamed endothelial cells, making the neutrophils stick and they squeeze through the endothelial membrane in a process called diapedesis, exiting the blood vessel and attracted by chemoattractants such as the FMET peptides of the bacterial proteins, LPS of the bacterial membranes, and tumor necrosis factor. Pathogen recognition involves pathogen associated molecular patterns. And also, the polys will recognize opsonins. And also, antibody coated microorganisms are also recognized by the neutrophils. The bacteria is engulfed, ingested in a phagosome. A lysosome comes along and fuses, forming a phagolysosome. The neutrophilic granulocyte's weapons are its granules, primary xerophilic and secondary specific granules, which contain antibacterial enzymes and antibacterial proteins. Chemoattractants promote chemotaxis of millions of polys into the battle zone of inflammation, where they die off, eventually necrobiosis, forming the familiar pus fluid you may have seen in smelly body fluid specimens. Which leukocytes are capable of phagocytosis? We know that neutrophils and monocytes are professional phagocytes. Eosinophils, they can also be involved in phagocytosis in a different way. We see eosinophils attacking parasites, and we see them also elevated in when conditions of asthma and allergies in atopic individuals and the hypersensitivity reactions. But let's see how they kill parasites. Eosinophils surround the parasite and they will degranulate, killing the parasite. And that's how they accomplish their role in phagocytosis of parasites. Actually, you can see the parasite's too big to be engulfed by a single eosinophil, which is a true statement about specific secondary granules in neutrophils. They are eosinophilic, so they're not dark purple and they don't contain esterase. And they do help kill bacteria. So they appear at the myelocyte stage. They appear in the dawn of neutrophilia. That's the beginning of the appearance of specific secondary granules at the myelocyte stage. And you can see that primary granules appear in the promyelocyte, and myelocyte appears, they have secondary granules. Neutrophils have these weapons called neutrophilic granules, and the primary granules have myeloperoxidase enzymes plus other enzymes. The secondary specific granules has many antibacterial enzymes and other antibacterial proteins such as lactoferrin, which we'll see later. Did you know there are tertiary granules? They contain lysozyme plus other enzymes. Myeloperoxidase is an enzyme that makes hypochlorous acid, otherwise known as a bleach, and this will kill bacteria. And lactoferrin binds to the LPS of bacterial cell walls, and it is also antiviral. And it will sequester iron, which is needed for bacterial enzyme reactions, such as the cytochrome reactions. And lysozyme, it's very effective it hydrolyzes the peptidoglycan cell walls of gram-positive bacteria. In the peripheral blood smear exam, you can see evidence of this infection with toxic granulation. Also, you'll see vascularization and Dolly bodies in the neutrophils. There's our quick review of the neutrophil, and we see how it plays a very important role in the immune system. And these are references in case you want to have further study. And please like and subscribe, and see you in our next video.